For the introduction. Hello everyone, my name is Anshu Gupta. I am a second year PhD student in CSE department at UC San Diego, and now I'll be going to present my work on accurate, scalable, and fully automated inference of species tree from raw genomic assemblies using ROADIES. So let's start with a brief introduction about my work. So we are all in the era of phylogenomics where genomic data is widely being used to construct the species tree. And there has been a significant improvement in the sequencing technologies over recent years. And because of that, we see a huge number of genomic data of different species being available these days. So this graph basically shows the exponential increase in the number of genomic data over a couple of decades for almost all sorts of species. And this massive improvement in sequencing technologies has enabled multiple consortiums to sequence tens to thousands to even millions of species on Earth. And this sheer scale of data, which is available these days, has immense potential to solve multiple complex questions and debates which is there regarding the tree of life. And to give you an example of one such debate, let's take a look at the avian phylogeny. So this picture shows the different phylogenetic trees inferred from the bird species, and this is being taken from 4 different papers. There are multiple papers, of course, but this figure basically shows that overall the picture looks same. Overall, the relationships across the orders of the birds look same, but there are slight differences in a couple of orders, and this shows that still some of the orders in the bird species are hotly debated. And this shows how important it is nowadays to construct an accurate species tree, and also it will be crucial since we will have more and more data sets in near future. Now let's understand how species tree are generated from genomic assemblies. So there are multiple steps and multiple ways to do so. So there's one way to do is to perform multiple whole genome alignments. Now multiple whole genome alignments are computationally very expensive, especially if you're going to have large scale data sets. Also, there's another problem in multiple whole genome alignments is that you need guide tree as an input. Now, the accuracy of the guide tree governs the accuracy of the alignment, and also that depends, and that governs the accuracy of the final tree. So that becomes like a chicken and egg problem, and it's hard to resolve. There's another approach to do that, to infer the species tree, and that is basically to annotate the genes and then infer the orthologs, and then you estimate the gene trees and the species tree. Now, why there is 2 gene trees and species tree step? Because to basically to take care of the gene tree discordances. Now, what are gene tree discordances? Gene tree discordances happen because same set of genomes, different parts of, if we study the different parts of the same set of genomes, it infers different evolutionary histories. So as a result, there are differences in the gene trees and the species tree, and we need to take care of these discordances in order to get the accurate tree. So there are multiple discordance aware tools available these days, and one such example of discordance aware tool is Astral, which is widely being adopted in multiple bioinformatic pipelines. So Astral estimates the species tree by taking a bunch of unrooted gene trees. It's a summary-based method, and it is also statistically consistent method. Even though we have discordance aware tools, there are other challenges that still exist in the entire pipeline of conventional approaches. That is, first of all, it's very difficult to infer orthologs because there are also presence of paralogs. So inferring true orthology is challenging. And also gene annotation is also not very straightforward, as in you need to annotate the genes, and for that you need to have some domain expertise. There are some manual hand curation steps involved, and that makes the process error prone. And because of these challenges, there has been almost no such attempts to do the entire automation, like from starting to ending, without requiring any other data apart from the input genomic assemblies. There has been conventional approaches there, but those are not computationally feasible to do so if you have large-scale data sets. And considering these challenges, the objective of our work is to develop an approach to estimate the phylogenetic trees of various species. And that approach should be reference-free because we want to get rid of reference bias. That approach should be orthology-free because we want to get rid of the estimation of true orthology. That approach should be alignment-free because we want to get rid of this computationally expensive multiple whole genome alignments. And also we want to be discordance aware. Now, considering all these objectives, we develop our tool ROADIES. And this is the full form of this ROADIES acronym that fulfills all of these objectives. Now, what is ROADIES? ROADIES is basically a tool which estimates and generates the final species tree in Newick format. And as an input, you just need to give raw genomic assemblies. You don't need to give any guide tree. You don't need to give any multiple whole genome alignments, or you don't need to do any orthology inference. Now, what ROADIES does? How ROADIES is different from the rest of the conventional approaches? So there are multiple different things that we do. So overall, the idea of ROADIES is to do homology-based species tree estimation. And that improvement has happened because of the improvement in Astral. So Astral Pro is an improved version of Astral. This is also a discordance-aware summary-based species tree estimation method, similar to Astral, but this improvement has happened because it considers paralogs as well as orthologs. So this removes the requirement of the user to separate out orthology and paralogy and makes the overall process quite faster. And since we do homology-based species tree estimation, we also get rid of the orthology inference, and instead we do the homology inference, which is much more easier and faster. And also, instead of the gene annotations, we perform random sampling. When we say random sampling, that means we randomly sample small subsequence of the genomic sequences across the input assemblies. When I say the small subsequences, we treat it as genes, and that is not protein coding genes, which is usually considered conventionally. This is you can say coalescent genes or short recombination free regions. Now, there are multiple benefits of doing random sampling. The first benefit is that since we are not just considering and restricting ourselves to protein coding genes, we do consider intergenic regions or intronic regions as well, because they are neutrally evolving regions and they follow the assumption of phylogenetic tools better. And the second benefit is that since we are uniformly sampling these genes across the input genomic assemblies, we are removing the reference bias. And with this random sampling, we are also removing the requirement of multiple whole genome alignments. And because of these differences, our tool is quite fast, fully automated end-to-end, and it's quite less error-prone. Now let's talk about the implementation of ROADIES pipeline. So the entire ROADIES pipeline is implemented into 6 different steps. Now I'll talk about each of the steps in detail. So in the first step, we randomly sample a bunch of genes from the input genomic assemblies. And once you have the bunch of genes, then we try to infer homologs of each of the genes from the rest of the species by using the genome alignment, whole genome alignment tool LASI. And then we filter out the bad matches and also the repetitive matches. And then we perform the multiple sequence alignment of the homologous sequences, which is taken from each of the gene. Once we have the bunch of MSAs, then we do gene tree estimation of all those MSAs. Now, multiple sequence alignment we perform using PASTA, and gene tree estimation we do perform using RAXML NG. And then once we have this bunch of gene trees, at the very end, we estimate the species tree using Astral Pro. So this is the entire pipeline of ROADIES from start to end. Now, there's one problem with what we do. Since I mentioned that we need to sample some bunch of genes, we don't know how many genes to start with. So the optimal gene count differs from different data sets. Like for mammals, we had to take 4,000 genes in order to get the optimal species tree. But for birds, since it's a very complex evolved data set, and it has high level of discordance, so we had to go for like at least 64,000 gene count to get the optimal tree. So how do we know which data set, for which data set we need to have how many optimal gene count to get the optimal tree? And for that, we developed an adaptive algorithm where basically we iterate the ROADIES multiple times, iterate the entire pipeline multiple times. And the main idea is that with every iteration, we keep on increasing the gene trees. And with more and more gene trees, the trees become much more better, much more accurate. And we keep track of the confidence of the trees at every iteration. If the trees become confident, we stop. Now let's talk about the methodology, how we evaluated our results. So we picked 3 different data sets: mammals, flies, and birds. And for each of the data sets, we get the final species tree. Now, how do we know whether our final tree is as accurate or not? For that, we got the reference tree from the papers which discussed about these phylogenies and which are widely adopted in the scientific community. And we tried to match the 2 trees using normalized RF distance as a distance metric. And we also measured the confidence of the branches, which were highly debatable, using the local posterior probability and the quadrat scores. Now, this figure shows the final species tree that we get from the 240 different species of mammals taken from the Zoonomia project. So this figure is quite hard to view, I understand that, but I would like to emphasize that ROADIES was able to accurately group the species to its correct phylogenetic orders as per the reference tree. We also tried to see the order level relationships across all the species from the ROADIES on the right hand side and from the reference tree on the left hand side. As we can see, both the trees are highly similar to each other. However, there are certain differences between these two trees, and interestingly, these differences were only limited to highly debatable branches. So we saw a bunch of papers, and some of the papers supported our results, some of the papers supported the reference, and some of the papers came up with their own solution. So we don't know what's the correct answer to this, but I would like to mention that ROADIES was able to accurately capture the relationships, at least for those branches which are well settled. Then if we talk about the convergence, the left-hand side of the figure shows how ROADIES, the final species tree of ROADIES gets more and more confident with more and more gene trees. And also we see on the right-hand side how the normalized RF distance decreases. That means if the distance is less, that means tree becomes much more accurate and much more similar to the reference tree. We did the same sort of analysis with flies as well as the birds. I'll not go into detail that much, but I would like to mention that we got pretty accurate results for both the datasets as well. We also estimated the speed up of ROADIES compared to the conventional approaches. For that, we estimated the wall clock time of ROADIES from starting step to the ending step for mammalian dataset. And for the conventional approach, we took there's one tool called TOGA, which does the orthology inference. So we perform the whole genome alignment of a pair of mammalian genome and inferred the ortholog using that tool. And when we compared the wall clock time, we saw that ROADIES was almost about 176x faster than the conventional approach. And that shows the potential of ROADIES to speed up the inference of large-scale datasets. We also wanted to see how ROADIES scales well, how ROADIES scales if you have like more number of species and if you have like more number of cores. In the right-hand side of the figure, if you can see, ROADIES scales pretty well, almost like super linearly, but quadratically with more number of species. And as you can see on the left-hand side of the figure, this shows how ROADIES scales well with multiple cores. And this has been done only with single node multi-core environment. We are planning to scale ROADIES further on multi-node environment. So in future, we plan to expand ROADIES with multiple data sets from diverse evolutionary timescales. So far, we have only tested for mammals, flies, and birds data set, which spans around 40 to 150 million years ago. We are right now trying to do for fishes, which is also like evolved much earlier than that. In future, we will be interested to do for the bacterial data sets. And on the flip side, we also wanted to see the evolutionary relationships of data sets from shorter timescales. So I would like to summarize the talk by saying that we saw how conventional approaches of estimating species tree are not computationally feasible for large-scale data set. It has some manual approaches. It has orthology estimation, which becomes much more challenging in future. And we addressed all those challenges and developed an automated tool to infer species tree directly from the raw genomic assemblies without requiring any other input. And also, we were highly configurable, scalable, and we were able to get the accurate results from mammals, flies, and bird datasets so far. So with that, I would like to thank everyone for listening. I would like to thank my advisors, Yatish Turakia and Siava Shmirarab, for their continuous help and support, and the collaborators for their continuous feedback. ROADIES preprint is now available online along with the code and documentation. So I would be happy if you can test it out for your datasets, provide your feedback, and yeah, thank you so much.
Thank you. That was very interesting and impressive.
Thank you.
You showed us how topological comparisons. I mean, you don't have much time, but you showed topological comparisons in terms of trying to assess correctness. Did you look at branch lengths? I have two questions related to that. One is, did you look at branch lengths? And the other one is, if you look at fine structure rather than the backbone of the tree, what do you see in terms of topological correctness?
So we did check branch lengths, but we did not evaluate it and analyze that in detail. We would plan to do that in future. And also, so there has been a recent change in Astral because Astral was not able to support the branch length. So there has been a recent change in that tool, and we are planning to add that in near future. And for the fishes, but I would like to say that we were able to get with the newest Astral tool, which supports the branch length, since we are doing it for the fish data sets, we were able to get pretty good branch lengths. But there was some of the very long branch lengths, which we are eventually trying to see what's the reason for that. And also, I don't know what was the second question, but I think it was more like the finer details.
Topological correctness sort of in the close to the leaves as opposed to in the background.
So fortunately ROADIES was able to get like within an order for all the species, the topology was correct.
Thank you.
Yeah, thank you for your question.
So since you're picking loci randomly out of genome assemblies, is there any attempt to avoid highly repetitive elements? Is this masked, or are they fine to include?
Yeah, we do include that. So as I mentioned in the filtering step, so in the filtering step, we do mask repetitive sites. Thank you.
Hi, thank you very much for your talk. It's really interesting. You mentioned there's no reference bias and then you sample the genes randomly, but then if the input are the assemblies, where do the genes come from? How do you sample the genes?
Sorry, I didn't get you.
get
Can you repeat your question again?
Yes, sorry. You mentioned there's no reference bias and that you randomly sample the genes, but if the input is the assemblies, where do these genes come from? How do you find them?
So it's not like protein coding genes. It's like just random loci, it's random subsequences.
Okay.
It's like random region. It's not like protein coding genes specifically. It's not like annotated genes.
Okay.
Yeah. Thank you.
Thanks for the talk. I was wondering, did you look at how your method handle difference in quality in assemblies, mainly like does work well when the assemblies is really incomplete and really fragmented?
We did not consider the quality of the assembly so far. We had this assumption that the reference, like the data set that we get, are of good quality. So we have taken it from like Bird's 10K genome project, like for zoonomia. So these are like well-known projects with good quality. So we put that assumption. But yeah,
Thanks.
we need to take care of that in future. Thank you.